Welcome back. We are looking at the application of some transport principles in cutting edge research. In the previous class, we looked at the liquid phase oxygen supply system, liquid phase oxygen supply strategy details, and I showed you some details of the economics, whereby the cost is brought down to about 15 paise or even less per kilogram of oxygen. At that stage, our thoughts were something like this. Did we solve the classic problem of oxygen supply to bioreactors? It was a classic problem. Okay. Yes, because as you saw, oxygen was made available to the cell at the place of need, and whatever the oxygen does not need seems to come out of the cell, right? So that's as complete a solution as you can get. Yes, we did solve, but what I did not tell you very clearly was that this strategy affects the growth. Okay. Why? Because we are using hydrogen peroxide. Okay. Although hydrogen peroxide is made in the cell as a part of its metabolism, it is something called a reactive species, right? And a reactive species can affect the growth of cells, the metabolism of cells in very many different ways. And that is the reason why whenever we use this, although the per cell productivity was higher, the cell yields were lower in the case of LPOS. This we had already noticed, and we were figuring out means by which we can offset that. The thinking was, if we can somehow offset that, since the per cell productivity is higher, if the cell concentration is also brought up simultaneously, then the productivity will go up that much better. Okay, the productivity will become that much higher. That was the thinking behind that approach. Then when we started probing this was way back in 1995, we realized that we are adding hydrogen peroxide, there is iron in the medium, and you have the classic Fenton reaction that's happening, the reaction between hydrogen peroxide and iron to give you hydroxyl radicals and so on and so forth. And those reactive oxygen species, much more deleterious reactive oxygen species that are generated from hydrogen peroxide, could be the reason why the cell concentrations were low. Okay. As such, we are using low enough concentrations of hydrogen peroxide, but still whatever little is used, there is a deleterious effect that is cumulative, and that results in lower cell yields. Okay. And this is what we started looking into way back in 1995. The first PhD, my first PhD student, Manjula Rao, joined with a reactive oxygen species background in January 1996, and she started looking at this problem. Her approach was, her thoughts were, can we vaccinate against these reactive species effects? She started thinking in that direction. The idea was, we will somehow make the cells immune to the negative effects of reactive species, and thereby when we use the LPOS, the cells are more robust, and therefore they will not die as much, or their growth will not be affected as much. So this was her thought, and we did a lot of work in that. Our entire direction of research changed, of using LPOS or viewing LPOS from a reactive species point of view, and coming up with entire strategies based on reactive species themselves. Okay. It so happens that if you induce reactive species, this is what we found out when we went and characterized this, if you induce reactive species, you actually improve productivity by a totally different mechanism in the cell. Okay. That is not highly transport related, so although very interesting, I will not get into that in this particular lecture or in this particular course. Some background on reactive species for you. Many of you may not know what they are. They may have an unpaired electron, may not. They are very reactive. In comparative terms, all the way there is second order rate constant for hydrogen peroxide with iron is only 76 per molar per second. the hydroxyl radical rates are typically of 10 power 9, 10 power 10 per molar per second, the second order reaction rates. So, it spans a wide range. So, it is only by practice we will know what to call as, what to categorize as reactive oxygen species or not. So, the reactive species are general, whichever may have an unpaired electron and are reactive. The ones that are derived from oxygen are called reactive oxygen species. You could derive them from nitrogen, you could derive them from halogen, and so on and so forth. The ones that we focused on, which are most important in fact because of their reactivities, hydroxyl free radical. This is a highly reactive one. Okay, this is the most reactive one in fact. Hydroxyl radicals, OH dot, the dot is on the O, the unpaired electron is on the O. The superoxide free radicals, O2 dot minus. And this is nothing new. The reactive oxygen species have been implicated in very many different diseases. In fact, I have an entire course, an elective, on reactive species in medical technology, medical and related technology, right? And yeah, that's a whole different field altogether. So, this is what I mentioned, that I will not get into in great detail, that our whole direction changed when we started looking at reactive species, and we showed that to work in very many different systems and so on. Today's paper is on this aspect. Okay. We were looking at chemical stresses to this cell, such as hydrogen peroxide addition as a part of LPOS, HOCl addition to induce reactive species in the cell, and so on and so forth, which I have not discussed much in this course. So, when you add these, something happens. We wanted to see, we saw that the effects of a chemical stress are mediated through reactive species. We wanted to see whether the effects of a physical stress is also mediated through reactive species, because physical stress, shear in bioreactors is common. That is, we were working on bioreactors at that time, very relevant, and we wanted to see whether they are mediated by reactive species, and that is how we got into this. And as I very briefly mentioned, we had to come up with a means of growing cells at defined shear levels. We had used a device here, something like this. Now, let me switch to the actual paper. I have an entire paper to discuss. So, yes. So, this paper published again in 2003 in Biotechnology Progress, macro level and genetic level responses of Bacillus subtilis to shear stress. Okay, authors are Susmita Sahu, my third PhD student, Rajesh Kumar Verma, my MTech student, and all these are colleagues, A.K. Suresh, K. Krishnamurthy Rao, Jayesh Bellare, and myself. Okay. Here the spelling is different. So, here some parts of the introduction for you. Hydrodynamic or shear stress, everything is transport now, at sublytic levels significantly affects the macro level cell responses such as morphology, growth, and productivity. This is known, nothing new. The macro level responses result from altered genetic level responses to the shear stress. Although shear is normally perceived to be deleterious, it can also be beneficial, since shear affects cells at a fundamental level. A better understanding of its effects on the cells in culture could lead to the design of appropriate bioreactors or the operation of existing bioreactors in favorable regimes. Okay, this is way back, 17 years ago. 17 years ago this was published. So, the work was done 2 years before that. Studies on the effects of and cell responses to shear are available, predominantly on mammalian, plant, and insect cells. However, information on shear responses of bacterial cells, which are widely used in a variety of industries, including those producing high value products such as pharmaceuticals and other recombinant products, is not as abundant. The information on genetic level responses of bacterial cells to shear is not available. Okay. So, that is what we started. That was our motivation to get into this work. Many studies on shear effects have been reported in stirred tank bioreactors, in which it is difficult to decouple the effects of shear and oxygen transfer. Everything is transferred that we have seen here. Okay, effects of shear, oxygen transfer, KLa. For example, increase in RPM to increase shear rate would also result in an increase in the volumetric oxygen transfer coefficient. Okay. Further, it is difficult to quantify the shear levels in the stirred tank, because why? We have already seen the velocity profiles are completely undefined, right? Difficult to measure also. And we know that shear stress is based on the velocity gradient. Therefore, you have the velocity profile, you can figure out the shear stress profile. And the average shear may not be representative of the actual range of shear levels to which the cells are exposed in the vessel. However, shear studies in a Couette flow regime, which is achieved in the space between two concentric cylinders, one of the ways of achieving that, rotating at different angular velocities, are available. Although the defined and uniform shear field in Couette flow facilitates analysis, it is non-trivial to supply oxygen to aerobic cultures at adequate rates without affecting the flow field for the entire duration of growth. Okay, this was the challenge that we contributed to overcoming. One is therefore constrained to use devise short-term experiments, or to introduce oxygen air in a manner that might compromise the defined shear field. There are some details there. In this work, the Couette flow bioreactor has been designed and fabricated to cultivate cells under well-defined laminar shear conditions for the entire duration of growth. Okay, this was our contribution. Earlier, you could do for short times, as long as oxygen did not become limiting. Till that time, you could do, but not complete cultivations. The CFB, the Couette flow bioreactor, was designed to ensure operation in the Couette flow regime by using a thin annulus, as well as to ensure oxygen supply without compromising the laminar flow profiles, so that long-term cultivations of an aerobically growing organism can be carried out in a defined shear environment. The problem of oxygen supply with minimum disturbance to the flow field was addressed by having oxygen inside the hollow inner cylinder and transferring it to the annular space containing the culture through an oxygen permeable membrane. Okay. So, let us describe this here. Okay. This is the longitudinal section of the Couette flow device. You can see the inner cylinder here with a tapered end about half a degree or 1 degree or something like that, and you have the outer cylinder here, and of course, this is a bearing to ensure appropriate rotation and so on and so forth. There is an auto bearing here to ensure a maximum of 1500 RPM here. So, this is the space, the annular space where the culture is grown. Okay. How do you supply oxygen to this so that you could carry out cultivations over the entire length? What we did was, we cut holes into the surface of the inner cylinder, okay, and then covered the outer surface of the inner cylinder. In other words, the surface that faces the culture with a Teflon membrane. Okay. So, the oxygen that is provided here will pass through these holes, that is not a problem, pass through the membrane that is covering the holes and get into the annular space where the cells are grown, and thereby it supplies oxygen continuously to these cells. And then we had to characterize it and so on and so forth. We found that it can provide oxygen to significant levels, required levels by the cells, and then we had to adjust the cell concentrations and so on and so forth. Okay. So, this entire thing is nothing but all transport, right. We had to, for example, here let me show you this. Where was it designed here? Yeah, here, right here. Okay. Shake flask cultivation. Okay. Uses control. Two types of devices have been used in the present cultivations. The cuvette flow bioreactor at several speeds of rotation to vary the shear rate and the shake flask. For a meaningful comparison of the two devices, we need methods of calculating shear rates in these two devices. The shear rates in the CFB can be exactly calculated from the velocity field. You know, we derived this in this class. We derived the exact same expression in this class. Okay. And that, you remember this gamma dot, I would have used a different symbol here. The shear rate gamma dot function of r to omega kr by r squared k squared minus 1. This we had actually derived in this class, and this is, we are using this in research, right. Look at the direct application of something we derived in an undergraduate course being used directly in research. Okay. So, that is the power of it. And then of course, there are various other aspects of transport, Reynolds number, power number, here KL effective, which is the, we know the KLA. So, KL can be estimated separately, and so on and so forth for this case. So, for all that we have these expressions that have been looked at, derived, and so on and so forth, or taken from the literature. And then this was used to get some very interesting results. Okay. So, here one result, let me show you here and then go back to the presentation to show you the other results. This is the SEM, the scanning electron micrograph of the cells that are grown in the shake flask. The cells that are grown in the shake flask with minimal shear are nice and long, right, you see here, whereas that are grown in the CFB at 1500 rpm or 1482 second inverse shear rate is much smaller. Okay. When you employ shear, you expect the cell to elongate and so on and so forth. Okay. Here what we find is completely opposite, completely counterproductive. This is a very interesting result for us. Okay. And there were other interesting results. Some results, let me show you as in that presentation that we were looking at. Let me close this now. Right. So, here this is the reason why we got into it. We wanted to look whether the reactive oxygen species mediate the effects of physical stresses. Also, we designed and fabricated a device entirely based on the principles of transport phenomena. We had used the expressions that were derived in this course, and such a device could do entire cultivations under defined shear stress. It can decouple oxygen and shear effects and so on. And these were some of the very interesting results. You see specific intracellular ROS at a certain point versus shear rate. Okay. As the shear rate increases, the specific intracellular ROS increases, the reactive oxygen species. The effect of shear is being mediated through the reactive, is possibly being mediated. We have actually shown it is mediated. This data shows that it is possibly being mediated by specific intracellular reactive oxygen species. Then we looked at the growth, the cell concentration profiles, cell concentration versus time. With the control, it is this green dots, which is standard, okay, in the shake flask. That when it was grown at 1482 second inverse, look at the increase in cell concentration, maximum cell concentration, and the growth rate. You could calculate the growth rate from this, you know how to do that. So, both were different, right, much higher when the shear was employed. And also the specific intracellular enzyme level of 3 different enzymes. This is extracellular, this is intracellular enzymes. And with the shear rate, there are changes in 2 of these enzymes. Okay. So, all these were interesting, and then Susmita also worked out the mechanism by which this happens, and she proved some parts of it. What she showed was the shear stress activates our shear stress, right. It activates the NADH oxidase, which is an enzyme on the membrane. So, NOx NADH oxidase gets activated. That causes the induction of reactive oxygen species such as superoxide and superoxide derived radicals. And then a lot of molecular biology happens here. There's a sigma B transcription factor that gets involved, and so on and so forth. So this stress response, in other words, a lot of responses to the stress get manifest, such as changes in growth and other phenotypes of the cell. And one of those phenotypes is increased production of products of interest to us. Okay. So that is what was shown as a part of this paper. We saw the application of transport in this, and in the next class I think I will present probably a couple more and maybe finish with them. What do you say? Yeah, I think maybe 2 classes or 1 class, depending on how it goes. We will take up 2 other examples of this, and one will be pretty much current, the work that was published recently, and so on. See you in the next class. Bye.